We're about to start in about a minute, maybe less. Well, thank you for all being here this morning. But before we start, please be aware of the exit signs in case we need to leave the room on short notice. And for those that are on the internet, please be aware that for the Q&A, if you want to send in your questions, please do it at your earliest opportunity so we can have time to take it on the air. So with that, Eileen.
Thank you. I'd like to welcome everyone here in this room and those on the webcast to what I think will be an extremely interesting seminar that has 3 experienced scientists speaking today. So you're really getting a lot of information. The first speaker will be Professor Alan Goldstein from UC Berkeley. Alan has been a professor in the Department of Civil and Environmental Engineering and in the Department of Environmental Science, Policy, and Management. He was division chair of that second division. Professor Goldstein is also currently the co-chair of the International Global Atmospheric Chemistry Program. And his interests include biogenic emissions, atmospheric exchange of radiatively and chemically active trace gases, and he has been involved in a number of important field studies. Our second speaker is Dr. Mark Fisher. He is a staff scientist with the Atmospheric Sciences Department at Lawrence Berkeley National Lab and associated scientist at Cal State University East Bay. Dr. Fisher's research focuses on measurement and modeling of human ecosystem atmospheric processes involving trace gases, with an emphasis on the drivers, responses, and feedbacks to global change. And our final speaker is Abhinav Guha, who is currently with the Bay Area Air Quality Management District as a senior air quality engineer. His primary job responsibility there has been to conceptualize and institute a greenhouse gas monitoring and measurement program across the Bay Area. He worked on the data that you will see as part of this presentation. Thank you for joining us, and I think you will find this an extremely interesting presentation. Professor Goldstein.
Thank you, Eileen, for that kind introduction. And thank all of you for coming today, those of you who are here in the room, as well as those of you who are watching on the web. I appreciate getting your attention. We'll be talking about source speciation of greenhouse gases, non-CO2 greenhouse gases in the Central Valley of California, using in-situ measurements of volatile organic compounds that we've done over a few-year period at the Walnut Grove Tower. And in addition to the folks who are going to be speaking with me today, I want to acknowledge that the California Resources Board has paid for this work under a contract, and we appreciate that support very much. And a special thanks to Ash Leshkari, who was our program manager, and also people at the National Oceanic and Atmospheric Administration, Arlene Andrews and her team, who have been maintaining some of those greenhouse gas measurements at the Walnut Grove Tower for a long time now, in coordination with Mark Fisher, and also Dave Bush from TMB Systems, who's been maintaining the ozone measurements at that site for many years. So the overview is that we're focused on investigating the emission sources of methane and nitrous oxide in California's Central Valley and their seasonal variability. And we did these measurements from summer 2012 through early fall 2013. We did year-long measurements of greenhouse gases, carbon monoxide, and a suite of volatile organic compounds at the tower. And the goal was to separate them into seasonal periods and then try to understand how contributions from different source categories were changing as a function of season. And that's largely going to occur because many of these sources are biological and they have seasonality, both due to the anthropogenic management and the biological processes that are involved. We use 2 separate independent methods to try and assess this. One is called positive matrix factorization, using this suite of tracers, which can tell us about the sources contributing to methane and N₂O, and the other is inverse modeling to explain the N₂O and methane observations at the site. So in today's presentation, I'll be giving the overview, talking about the objectives and our approach. Then Mark Fisher will talk about the inverse modeling analysis and results, and they had started this work before the current project, and they've been doing this for quite a few years, and so I wanted Mark here to represent the work from his team. And then the PMF analysis and comparison with the inverse modeling and the current inventories, Abhinav Guha will talk about. And now Abhinav is currently at the Bay Area Air Quality Management District, but when he did this work, he was a PhD student in my group at UC Berkeley. But particularly given his new role at the Bay Area Air Quality Management District, I want you to hear about the work directly from him. Then I'll come back and talk about our conclusions and also say a few words about future research opportunities and needs. So as an overview of the current situation with California's greenhouse gases, as you all know, the Global Warming Solutions Act of 2006, AB 32, aims to reduce the greenhouse gas emissions from to 1990 levels by the year 2020. And as I'm sure you in this room and on the web are all aware, there's discussion of extending that to 2030 right now. I won't be talking about that in detail, but clearly this is an important topic for the state, and understanding what our inventory is really critical. And then being able to see how that inventory is changing with time to make sure that we're meeting these targets. So the ARB has internally developed a statewide greenhouse gas inventory for California using a combination of emission factors, activity data, and emissions modeling. And so sometimes I'll refer to that as a bottom-up inventory. So it's looking at the specific sources, emission factors, activity data, emissions to get an assessment of what the total is. That current inventory has methane as 9% and N₂O as 3% of the total greenhouse gas emissions in California. So as we reduce emissions to get back to 1990 levels by the year 2020, the expectation is these emissions would decline with time. So for N₂O and methane, emissions are actually dominated by area sources that are biological in nature. They display temporal and spatial heterogeneity, and hence can be somewhat difficult to assess and measure. And so what we set out to do was add additional observational data and high-resolution modeling estimates to try and take a top-down look at explaining the enhancements in concentration of methane and N₂O that are observed in the Central Valley of California, and to see if we can determine, looking from the top down, what the emission sources were, and then see how that compares to the inventory from the Air Resources Board. And also, we want to create a baseline of observations that hopefully over the next 20, 30 years, as emissions continue to decline, we can go back and compare new measurements to, right? So it's not just about doing this as a snapshot at one time, it's actually setting a baseline that we can compare to in the future, a baseline based on measurements. Okay, so California's current methane emission inventory taken from the CARB greenhouse gas inventory tool in November 2015 suggests that 29% of the methane over the state of California is from enteric fermentation in animals. Then management of the manure from those animals is another 26%. Landfills is about 20%. Then there are smaller contributions from wastewater treatment, natural gas pipelines, fugitive emissions, and rice cultivation that together make up another roughly quarter of the total emissions. So the big sources here, landfills, and then animal agriculture, enteric fermentation, and manure management. If we look at the same inventory for N₂O, we can see that about 2/3 of the emissions are assigned to a category called soil management, which is basically agriculture and urban setting use of fertilizers and then biologically mediated processes that are producing N₂O from the applied nitrogen. The transportation sector is shown in the California N₂O emission inventory as being responsible for about 12% of the N₂O. Then manure management is 12%. Wastewater treatment, electricity generation, industrial emissions add another 11%. So when we do measurements in the Central Valley of California from the tall tower that I'll be talking about, those measurements are specific to the region that actually influences concentrations that are observable at that tower. And Mark Fisher will be talking more about that footprint and what the transport of air looks like in that region. But in order to think a little bit more about what we're going to be able to compare our measurements at the Walnut Grove Tower site to, we have to look a little bit more deeply into the emission inventory for that specific region. Now, California doesn't have that region-specific inventory broken down through the ARB. We actually did that ourselves. We calculated the annual inventory based on source distributions for the Central Valley region surrounding the site, and that's basically the San Joaquin or the Sacramento area and the regions that are slightly upwind. Mark will talk a little bit more about those regions, but we call them regions 3, 7, and 8, and those are basically airsheds that are defined by the Air Resources Board. So based on the CalGEM 2008 methane sources, in that region, about half the total emissions should be from the dairy and livestock, with another 8% from rice cultivation, 20% from waste management, 17% from oil and gas, and a little bit from wetlands. For N₂O, about half the emissions are from agricultural soil management, with roughly 19% from manure management, another 13% from transportation, and then another 13% from industrial activities, 6% from wastewater treatment. So these regional emission estimates are a bit different from the state, but you'd expect that in the Central Valley of California, where there's an enormous amount of agricultural activity. And there are also, as we'll see in a little while, significant oil and gas fields in the area, for example. Okay, so our objectives are really to focus on assessing the seasonally changing apportionment of methane and N₂O sources in the region by looking at observations of those gases, N₂O and methane, and gaseous tracers that can indicate their sources. So that's really the big innovation that we tried to do in this study, is to identify some tracers and measure them simultaneously with the methane and N₂O to use them as indicators of where that methane and N₂O is coming from. So we added this continuous measurement of a suite of volatile organic compounds and nitrous oxide to the ongoing measurements that were at the Walnut Grove Tower, which had included CO2 and methane and carbon monoxide. And then we analyzed the mixing ratio time series for the greenhouse gases and the volatile organic compounds by a statistical source apportionment tool called positive matrix factorization that Abhinav will define for us to look at seasonality of these sources. And then Mark's group separately performed inverse modeling of the methane and N₂O enhancements observed at the site, and then we compare them to the emission inventory. So the site, the Walnut Grove Tower Study site, is just south of Sacramento by about 30 miles. It's at the western edge of the San Joaquin Delta in the Central Valley. And the site has a tower on it that was a communications tower. It's a really tall tower, 525 meters. There's a couple of pictures of it to give you some perspective. And having a tower that sticks that far up into our atmosphere in the Central Valley is really, really useful, because sometimes the tower goes above the inversion layer, so we can actually see a differentiation between the emissions from the sources that are adding trace gases to the air in the Central Valley and the background air that's coming from further away. And I'll show you a little bit of that. So here is just a little bit of the data, specifically the methane and N₂O data that were measured at two heights above the ground, 91 and 483 meters. And in the lower left, what you see is average diurnal cycles of the methane measured at those two heights, where the red is at 91 meters and the black is at 483 meters, and they're broken out by seasons. And what I want you to see there is that you very clearly see a much different diurnal cycle in different seasons, and that you see much higher concentrations at 91 meters than at 483 meters most of the time. And so we think of those differences in concentrations as enhancements, and it's those enhancements that really inform our modeling work and the PMF analysis. And so if we look on the, and that the seasonal variations in the emissions and the meteorology are both affecting our signals. If we look at the figures on the right, you can see the full annual cycle of methane on the top, and in red is shown the background concentrations that are derived from data collected by the team at NOAA from a variety of sites that are defining what the background over the Pacific looks like that's flowing into California. And then what you see are the daytime measurements of enhancements in at the Walnut Grove site. And so the difference between that red line showing the background and the enhancements are what the inverse model is trying to explain. Okay? And you can see that the enhancements are much larger in the winter than they are in the summer, and that's largely due to meteorology. So we have a lot more dilution in the summer and less dilution in the winter, and that's not because the emissions are higher in the winter, but because the dilution is much slower. And then for nitrous oxide on the bottom, you see the same sort of story. So you can see the magnitude of enhancements that we're trying to explain. So the setup for measuring methane, CO, et cetera, was already in place when we decided to try to add volatile organic compound tracers to define these different sources. And so this is a picture of the system that we set up. There was an existing set of inlets at these 5 heights, 525 meters down to 10 meters, where ozone had been measured for years by Dave Bush under contracts with the Air Resources Board and I believe the local Air Quality Management District. And so to those inlets, we added measurements by proton transfer reaction mass spectrometry. And I won't go in detail about how this instrument works, but it allows us to look in real time at a series of different volatile organic compounds. And so we actually were switching between these inlet heights every 2 minutes. And so in 10 minutes, we'd get a vertical profile. And then we typically average that vertical profile over a period of an hour. And so over the course of the year, when we did these measurements, we have average vertical profiles every hour. And before we actually started this project, we wanted to show the Air Resources Board it was going to be worthwhile. So we did some pilot measurements back in the summer of 2011. I'm going to show a little bit of information from those pilot measurements. So I won't go through this list in detail, but I want to say that the proton transfer reaction mass spectrometer is really measuring mass to charge ratios, and we associate those mass to charge ratios with specific chemicals, or sometimes it's a sum of different chemicals. And these are a list of the 20 mass to charge ratios that we measured, and an indication of what sources contribute to them. So for example, mass 33 is methanol. And methanol can come from livestock, manure management, vegetation, biomass burning. If I switch down to mass 79, 79 is benzene. And so benzene comes from fugitive emissions, it comes from gasoline, and also comes from biomass burning. And so when you get a suite of these chemicals rising and falling together in the observations, it gives us an indicator of what types of sources are contributing at that time. And then we can compare that to the changes in the methane and N₂O to see if they're changing together. So this is what some of that data looks like. This is averaged over our pilot observations, and I know this is a lot to look at, but let me talk you through a little bit of it. This is the period of our pilot observations in the summer of 2011. In the upper left here, what you're seeing is methane that was measured at 30, 91, and 483 meters. And this is taking data over about 40 days and looking at the average diurnal cycle. And so what you can see is the red colors are high concentration, the blue colors are low concentrations, and you can see sort of background levels up above the tower, particularly at night, and then you see these enhancements near the ground, and you can see there's a specific time of day typically when you get large enhancements, right? And then we can compare that to some of the other tracers. So for example, carbon dioxide is enhanced mainly at night, and this is when the boundary, the vertical mixing is slowest, and when vertical mixing is fastest, you don't get much enhancement in CO2. Carbon monoxide you can see is enhanced here roughly 3 in the afternoon all the way through the vertical profile. And interestingly, and Abhinav will come back to this, acetonitrile, which is a tracer of biomass burning, was highly enhanced at the same time as carbon monoxide. And you can also see it enhanced at night in the residual layer above the inversion. And this is because during that period in summer of 2011, there were a whole bunch of wildfires going on in the States. And so we can really see that beautifully. That affected the acetone concentrations. I'll just say a little bit more about some of these tracers now. Carbon monoxide is a good tracer of fossil fuel combustion, biomass burning. I talked a little bit about methane and CO2. Acetonitrile is a good tracer of biomass burning. But then we have tracers like toluene and benzene that tell us about gasoline and fugitive emissions. Turns out benzene also comes from biomass burning. So if we see benzene, carbon monoxide, and acetonitrile together, that tells us it was a biomass burning signal, right? If there's no acetonitrile in it, then that means that the CO and benzene probably came from fuel combustion, right? We've got some other really nice tracers like isoprene is a natural biogenic emission that comes mostly from the oak in the foothills surrounding the Central Valley, a little bit from plants that are in the valley. We've got acetone that comes from livestock, rice, vegetation, photooxidation. Acetaldehyde comes from similar sources, but also can have a significant source from biomass burning. And so I think you can start to get an idea that we've got a series of tracers that can help us differentiate sources. So if I go back now to this figure, you can see that the methanol cycle looks very different, for example, from the benzene or the toluene. Benzene and toluene are really high at night, and then as soon as the inversion breaks up in the morning, you see much lower concentrations. So this is indicative of fairly local sources contributing benzene and toluene, but the methanol is clearly not being enhanced as much at night. It's being enhanced more during the middle of the day, and it's all the way up through the vertical profile. So it's those covariances that are really giving us information about what might be contributing to methane at any given time. Okay, so we get lots and lots of data from this. We've got data at multiple heights for multiple tracers, and it can be a lot to look at. And that's sort of what I want to show you on the left. But if we look in a little bit more detail at some specific events, we can learn a lot. And I'm going to show you just a few individual events to give you an idea. So we are measuring temperature at all those heights on the tower we were. And so you can see I've converted those into potential temperature down here. When the potential temperature is the same at all heights, that basically means that things are well mixed. When the potential temperature shows a gradient and the potential temperature at near the ground is much lower than up top, that's when we have an inversion. And so we can see where we should get vertically mixed and where we should get inversions, and then we can look at what that does to the concentrations we observe. So in this example, we're showing isoprene and one of its oxidation products, which is methylvinyl ketone plus methacrylate. And you can see they have very different patterns. The isoprene has this really big spike that occurs around 6 p.m. And that happens right when the inversion starts to set up. So as soon as you get an inversion, local sources of isoprene are being trapped in that lower layer just right near the ground, and they're only getting to 10 meters. They're not even getting up to the 131 meter inlet. At the same time, those isoprene oxidation products aren't enhanced at all during the day. They're only enhanced at night, and they're only enhanced at the upper layers. And that's because what we're seeing is air coming downslope into the Central Valley of California from up in the Sierras, and it's carrying a bunch of isoprene oxidation products from isoprene that was emitted in the hills. It's going over the top of the Central Valley at night and then being mixed down in the morning. And I actually think that has really interesting implications for the air pollution issues in the Central Valley. And in fact, isoprene oxidation products from the surrounding foothills, emissions of isoprene does get down into the valley in the morning. But this is just to give you an idea of the power of having these measurements in the vertical profile. Now, if we look at a few enhancement periods where we saw enhanced methane, I want to walk you through what we could observe. So on the left is an example where we saw an enhancement of methane that also showed an enhancement of CO2 at the same heights, right? You see the same vertical pattern where this is closest to the ground, and then in the middle of the tower and the top of the tower. And carbon monoxide was enhanced in the same manner. Methanol wasn't. Acetonitrile wasn't. So if methanol wasn't enhanced, that means it's not coming from a dairy. If acetonitrile wasn't enhanced, that means it's not coming from biomass burning. But benzene and xylenes were enhanced. And so you can see that consistent pattern for methane, CO2, CO, benzene, and xylenes points to a fossil fuel source contributing to methane at that time. Here, in the next panel, we think this is likely a dairy source because we get this enhancement in methane that's associated with an enhancement of CO2 with essentially no enhancement of carbon monoxide, so it's not a burning source. But there is an enhancement in methanol that looks just like these. There's no acetonitrile, so no biomass burning. There's no extra benzene or xylene, so it's not from an urban or fossil fuel related source. Okay? And so we can differentiate that source, and here's another one where you get a big enhancement in acetaldehyde, CO, methanol, acetonitrile, and benzene. And we believe the only significant place we can get acetonitrile is from biomass burning. So this is clearly a biomass burning plume, and it doesn't have much enhancement in methane. And so it shows us that there's not a lot of methane coming from the biomass burning, but there may be just a little bit. Okay, so that's the background. And now, in order to make sense of this complexity of data, we need to take an approach that uses statistical tools to look for these patterns in a much broader way and understand their temporal variability. And so there are a couple ways that we're going to be doing that. And the main way that Abhinav will talk about, which is positive matrix factorization. But before that, we want Mark to talk about some of the meteorology and the inverse modeling and the sources in the region. So I'm going to turn over to Mark right now.
Good morning. Thank you. Thank you for excellent introduction, Alan. I'm going to run through some slides that describe the meteorology and our inverse model estimates of emissions from the Walnut Grove Tower for the Central Valley and Bay Area. This first slide shows potential sources of methane and nitrous oxide to the Walnut Grove Tower. I'm going to use the cursor to indicate the Walnut Grove Tower as this red symbol with yellow sources from the natural gas fields in the Rio Vista and some gas handling facilities. And then to the east of the tower and south of the tower, numerous dairies that are present in the valley. Over to the right on this slide, there is a picture that shows the land cover in the Sacramento River Delta and southern Sacramento Valley. You can see in the Delta there are wetlands, and in other parts sort of a mixture of agriculture. Let's see, I guess I go forward with this. Oops. in order to understand what we see at the Walnut Grove Tower, it's important not only to know where the sources are, but where the winds are bringing emissions to the tower. And this varies with season and time of day. And so I'm going to start just by saying that in the summer, everyone is probably very familiar with this, the very strong heating in the Sacramento River and Sacramento Valley area brings buoyancy to the air, which then draws in marine air through the Golden Gate and through the Delta into the Sacramento River. And conversely, at night, there's a downslope flow that Alan mentioned that brings air back from the valley through the Sacramento River area. And so there's this sloshing of air back and forth during the summer that's quite strong. In the winter, that less sunlight, less heating, and more sort of synoptic flow bias in the winds leads to sort of north-south flows. There is still some east-west, but much more coupling of the tower to predominantly Sacramento and northern San Joaquin air masses. Okay? In order to understand both the source apportionment done with the PMF that Abhinav will talk about and the inverse modeling that I'm about to show you some slides for, we divided the year of observations into 7 different periods that are both varying within the seasons and throughout the year. We did this so that we would be able to both discriminate sources and have periods when there were continuous measurements of all the different species that are necessary for the PMF analysis. And this table just gives you a feeling that there are a couple different seasons for summer, a couple different seasons for fall and winter and spring. Okay? Giving you more detail about the winds that we observed at Walnut Grove during this campaign, we have wind roses. These show the wind speed fraction, in other words, how often was the wind blowing with a given wind speed and from what direction. For this slide shows daytime winds for summer, early fall, late fall, and winter. And also then winter, spring, spring, and summer. And what you see is that, for example, I will use the cursor, the summer of 2012 looks fairly similar to the summer of 2013. There aren't big changes from year to year, at least for those periods. And then, generally speaking, the winds have more sort of north-south flow in the winter and winter-spring periods. When we look at daytime versus nighttime, this is now the nighttime slide, you see that the pattern, I'm going to flip back and forth, I hope this doesn't cause discomfort. You get a feeling that many of the patterns are reproducible, but it is more variable between day and night in the winter and spring, for example. This slide here, went the wrong way. So this gives you a sense of seasonality and diurnal variations in the flows, together with where we expect the sources of emission. It's possible to make some conjecture about the relative contributions of different sources in different seasons and different time periods. Okay? Now, I'm going to give you a few slides on inverse modeling, and this is effectively going to be a numerical integration of the measurement data, which is up here, with predictions of signals that we would expect based on the combination of our what we call a priori or bottom-up model. This is, in this case, the CalGEM model for methane, with footprints describing how sensitive the tower is to emissions across the landscape for a given time period. And what we do is combine that with the measurements in an inverse Bayesian, in this case, estimate that scales these a priori emissions to give us an optimal match between the measurements and the predictions. So it's effectively, in some sense, just a very large least squares model, but it's a least squares model that is weighted not only with our uncertainty in the measurements, but also an estimate of our uncertainty in those a priori emissions. The ultimate result is what we call a posterior estimate of emissions, which we can then compare with the prior model and see whether there are places where it agrees or disagrees. I'm now going to step through the steps of the process in terms of estimating those footprints. We start with a meteorological transport model that is effectively integrating our knowledge of, in this bottom slide, you can see time evolution of the height of the boundary layer, that is how deep the mixing into the atmosphere is as a function of the time of day, and the direction of the wind. Together, those give us a sense of where the air reaching the tower or that footprint is defined. And we're using the NCAR Weather Research Forecast model to do that prediction in nested grids that cover the western US, California, and then there is a very fine grid over the Bay Area that is used as well. To give you a better sense of how we estimate the footprint, we take that wind from the model and propagate particles backward in time from the tower at the time of a given observation and look, where are the particles in space? Are they within the boundary layer here, or are they out of the boundary layer in what we call the free troposphere, and therefore not coupled to the surface? We take the number of particles in the boundary layer above a given land surface element and the time they spend there, weighted by the height of the boundary layer to estimate the strength of the footprint. And we can do this at varied times backward, and this bottom left figure shows particles leaving some site and propagating backwards. Initially, for a few hours back, they're close to the tower. As they go further back, they get further from the tower. Here are examples of the footprints calculated for the Walnut Grove Tower in each of the observation periods of this measurement campaign. They vary with season in rough sense, directly related to the direction that the wind is blowing, how hard it is blowing, and the depth of the boundary layer within those seasons. What you can see is that in the summer and spring seasons, there is a preponderance of flow from the Bay Area, and then as you move from fall into winter, you get more Central Valley influence with less coupling to the Bay Area. One thing that's important to mention is that our certainty on these footprints, which we develop through a comparison with meteorological measurements from wind profilers in the valley, is on an individual hour-by-hour basis fairly uncertain. We don't believe there's a bias in those measurements, in that prediction of footprint that's strong, but we do have uncertainty because we don't know exactly which way the wind is blowing from the WRF model everywhere in California. And so we take that into account in the inverse model process. Using the footprints, we combine them with an a priori model of the methane emissions, and this upper slide here shows the CalGEM model for methane emissions. The color scheme is such that the blues are in the valley are, oops, I can't find the cursor there, are relatively strong. There are a few sort of violet sources that are extremely strong that you can't really see. Those are landfill pixels where there's a very strong emission in a very small area. And you just don't see them here because they don't show up on a spatial map of this scale. For N₂O, we did not develop an independent spatial model, and we're using the Edgar N₂O emission maps also at roughly 0.1 degrees or 10 kilometers. For the CalGEM map, we calibrate the map effectively by sector to the ARB's inventory at the state scale, and we assign a relatively large uncertainty to the emissions from any individual pixel because we aren't that confident about the sources at a very fine spatial scale. Now, I'm going to walk you through the posterior emission estimates based on the measurements and this prediction of signal. This slide shows the central California area here with the Bay Area sort of in purple, the Sacramento Valley, and the northern San Joaquin Valley, where we expect the footprint for this tower dominates. The slides on the lower left show for either different source types, crops, dairies, landfills, other livestock, natural gas, petroleum, wetlands, and wastewater, the source categories we have, the relative strengths, and I just want to emphasize landfills and dairy dominate the prior model and the posterior model. If we instead just lump all the source sectors together by region, we find that there is, in the prior model, very hand-wavy, roughly similar total methane emissions from each of those three regions. And in the posterior model, they are varying around with some degree of uncertainty, but with some seasonal signal. And I guess I would point out the one that shows up most strongly is in region 3, which is the Sacramento Valley. In summer, we get significantly more than the annual average, and we believe that that's due to rice emissions in the northern Sacramento Valley. So this is sort of an example of an imperfect but initial estimate of where emissions are obtained for different seasons. And if we take all seasons, average them together for each source, and then sum that, we end up with emissions that are roughly comparable to the existing inventory that was developed at that time, where the range of values was between 1.1 and 1.5 times the prior model. So roughly consistent or slightly higher than the existing inventory. Note that the contributions from the specific source sectors here are entirely based on the prior model, and we cannot, using only methane alone, uniquely ascribe to any particular source sector. Now, having walked you through that slide for methane, I'm going to repeat the same idea, but for nitrous oxide. Here, we find that we're using slightly different source sectors because nitrous oxide is contributed by a different mix. I mean, there is more influence from some of the other sectors, but in particular, ag soils really dominates most of this. The both in the prior and the posterior model. Here, the Central Valley really is the dominant source of ag nitrous oxide, and the simplest possible conclusion here is that that is obtained is both predicted in the prior model and obtained in the posterior model. Though again, the VOCs are going to be critical to tying that down better. I guess one thing I would say is that the results here are as with methane, but slightly higher than methane in terms of what we believe the total emissions are relative to the existing inventory, suggesting something may not be accounted for completely in the existing inventory, but at the low end of our estimates might still be consistent. So now I'm going to, having run you through what we can see from sort of essentially wind direction and total concentration variation measurements with time correlated with footprint. Abhinav is going to tell you more about how the VOC tracers define the source speciation. Thank you.
Thank you, Mark. Oh, yes. Many of you might have had some experience working with positive matrix factorization before, so I will just glean over some details. And so one of the reasons why we decided to use positive matrix factorization is because in our case, we don't know the number of greenhouse sources or impacting our receptor site. We also did not have an account of the individual chemical source profiles of greenhouse gas sources that are going to impact the site, and did not want to make any assumptions about the chemical speciation of those sources. But what we did have was concentration time series of a large number of coincidentally measured greenhouse gases and VOCs, which are ideal inputs to the positive matrix factorization method. What PMF is essentially is a receptor-only unmixing model which breaks down a concentration time series of a number of tracers into a mass balance of arbitrary number of constant source profiles contributing varying concentrations over the time series of the data set. And so as input to our PMF model, we have the concentration time series of all the measured tracers. And accompanying that matrix is an uncertainty matrix where we ascribe uncertainty to each of those individual measured data points. And on the output side, what we get is based on the number of factors which we assume, or number of sources that we assume to be influencing our analysis, a chemical speciation profile for each of those factors, and a time series, concentration time series for each of those factors, which can be combined to recreate that original concentration time series. And without going into too much details, let me show you kind of a visualization of how this works. Basically, we have the time series on the original time series on the left. We have a number of constant source profiles which do not vary, which are constant over the period of analysis, and a concentration time series of each of those source profiles, which in a net sum reproduces that original data set with a residual matrix as well, which cannot be explained based on the uncertainties that have been assumed in the analysis. So based on the PMF analysis which we performed over different seasons at Walnut Grove Tower, we could break down our analysis as being described by 7 factors. Not all the 7 factors were reproduced in each of the individual seasons, but in net sum we had 7 factors. And I'll go over the the constituents of these factors, but let me just lay out the sources that we see which influenced the measurements at Walnut Grove Tower, and the color scheme we have been using, which would follow in the plots and figures that follow after this slide. So we have the dairy and livestock source factor. We have a black source factor which we use, which we show in black color, which we called the urban plus oil and gas factor, and I'll attend to that in a bit, why we combine these two sources and call it the same as being a coincident source. We have primary biogenics and secondary organics, which will be shown in light green colors in the following plots. We have the agriculture plus soil management plus delta source factor in the maroon or purple color. We have a source factor which is related to isoprene emissions in light blue, and we have a source factor which is related to isoprene oxidation products in dark blue. And another factor which we'll speak in one of the slides is a forest fire or biomass burning related source factor. So let's move on. Before I talk more about the source profiles and the constituents of the individual source factors that we observe, I would like to show you a couple of plots of how the PMF output looks like for one season. Then I'll follow that up with description of all the source factors, and then I'll come back to all of these seasonal PMF plots for the other seasons as well. But this is how the PMF output looks like to us, and it's the analyst or the user or who ascribes a name or a source to a source name to a particular factor, and what we get is basically individual mass fractions contributing to that source factor, and those mass fractions add up to 1 in this case. And what you see for early fall 2012 is that 6 source factors seem to explain most of the concentration variation of the enhancements that we observed during that 1.5 month period. And what you see is in the early fall period, all the 6 PMF source factors were detected. The source factors contributions vary based on time of the day. If you look to the plot to the right, you see that if you see the plot for methanol, most of the methanol seems to be coming from the primary biogenics and secondary organic source factor, and the diurnal profile peaks during the middle of the day, coincident with primary emissions from the surrounding agriculture. But there's also some methanol contribution from the orange factor, which we call the dairy and livestock factor, which has a diurnal profile which replicates the diurnal profiles of primary emissions like that of methane, which also shows up in the same source factor if you look to the plot to the upper left, where you can see the source profile for the dairy and livestock source in orange. And this is basically the theme of the plots which follow after that. But before going to those plots, let me talk to you about how do we come up with the attribution of certain categories of sources to these individual factors which the PMF analysis produces. So dairy and livestock factor. In this factor, source factor, you'll see that methanol and N₂O were some of the primary constituents of this source factor, but besides methane and N₂O, this source factor also has significant contributions from methanol, acetaldehyde, acetone plus propanol, methyl ethyl ketone, and all of these oxygenated VOCs have been shown to be emitted in significant quantities from a variety of livestock and dairy studies that we reviewed. The presence of methanol in this particular source factor indicates that this source factor is biological in origin and not combustion or fossil fuel related. The methanol to methane molar ratios from this particular source profile conforms and matches very closely with the methanol to methane profile emission ratios from a variety of dairy chamber studies as well as airborne studies airborne studies conducted over dairy intensive regions in the Central Valley. Also the N₂O to methane molar ratio that we observed from our the dairy source profile in this analysis matches closely with a similar PMF analysis that was conducted as part of the Kalnick study in 2010, and the N₂O to methane profiles are very for this study is very similar to the N₂O to methane ratios in the dairy factor that we observed in Bakersfield during Kalnick's 2010. So that leads us to believe that the orange source factor is predominantly being dominated by dairy and livestock contributions. Moving on, the black source factor. Most of the CO, benzene, and toluene seen in this analysis was attributed to this black source factor. It also contains acetaldehyde and acetone. So the presence of these compounds suggests that the likely source of this factor is some sort of combustion or fossil fuel related contribution. When we look at the toluene to benzene ratios for this particular source factor, we see that the toluene to benzene ratio is somewhat depleted if you compare that to toluene to benzene ratios in fresh urban plumes which have a lot of transportation and vehicle tailpipe imprints. This suggests that this toluene to benzene ratio is, this source factor is not being dominated by contributions from the nearby Interstate 5, which is about 5-7 miles east of the site, but mostly from transport of urban plumes containing toluene and benzene, because toluene and benzene have a different photochemical depletion rate. So the toluene to benzene ratio decreases as these urban plumes are transported to rural areas, and that suggests that at least part of this part of contribution to this source factor is coming from the upwind urban core of San Francisco Bay Area. But while these urban plumes pass over the San Francisco Bay Area and reach Walnut Grove Tower, they also flow over croplands and some intensive natural gas fields in the Rio Vista region. So some of the contributions which we see in this source factor could be actually being added by local sources from the Rio Vista natural gas fields. Our review suggested that toluene-benzene ratios from natural gas fugitives is also very depleted and is very similar to the ratios that we observed in this source factor. So we really cannot pinpoint if this black source factor is completely urban and Bay Area specific, or if it is natural gas specific, or if it's a combination of both, since Walnut Grove Tower is mostly downwind of both these broader source categories. Hence, we decided to call this source factor an urban plus oil and gas factor, and look at and try to explain our observations in the context of these two source categories. To wrap up, acetaldehyde, I spoke about acetaldehyde and acetone being present in this source factor, and secondary production of acetaldehyde from photooxidation of light alkanes is well known, and its photooxidation of alkanes is also responsible for some contributions of acetone, and so that kind of reinforces our nomenclature for this particular source factor. Moving on, we detected strong presence of a primary biogenic and secondary organic source factor in all the 6 or rather 7 PMF seasonal periods. This source factor has heavy contributions from a number of oxygenated VOCs, and the diurnal and vertical profiles of that individual source factor over the time series of the individual PMF seasons suggest that the emissions from this source factors that source factor is occurring mostly during the middle of the day, peaking during the middle of the day and falling down rapidly during night time, something which is not very typical of long-lived primary pollutants. So we use this kind of observation to basically attribute this source factor to primarily to biogenic origins from the ongoing agriculture around the Walnut Grove Tower area. Also, during the contribution of this source factor to the PMF attribution is really low during the winter and the early spring period as opposed to the summer season, and this coincides with roughly the growing season of the agriculture season, the growing season. We also detect a strong a strong presence of a source where a lot of the nitrous oxide and monoterpenes is attributed to that source. Now, monoterpenes is primarily originates from agriculture and corn harvesting and other harvesting operations. And N2, as we know, one of the more dominant sources of N2O in California is is basically soil-driven, fertilizer-led soil-driven emissions. And so this is a case of co-located emissions. There are primary emissions taking place from plants and crops that are growing on the croplands, which have been heavily fertilized, and there's soil emissions of N₂O from the soil, but these emissions are occurring from broadly the same source region. And so when they, and hence the PMF detects these contributions from two sources as a single source factor. Moving on, we have isoprene, a very high isoprene dominated source factor, which peaks during the middle of the day exponentially and falls off rapidly during nighttime. We have an isoprene oxidation product source factor, which has a slightly different diurnal profile from the diurnal profile of isoprene, and the peak of the isoprene oxidation product source, the factor happens a couple of hours following the peak of isoprene, and that leads us to believe, and it's principally composed of methylvinyl ketone and methacrylene, which are isoprene oxidation products, and these show up separately as two distinct source factors, but not during all PMF seasonal periods. They only show up during the spring and the summer season. Actually, only the summer season. So now that I've kind of given you a flavor of how we get down to what we call individual PMF produced factors, let's look at some of the other seasonal periods. So late fall, as was middle of October to November 30, you see here that the temperatures start cooling down, and the PMF no longer produces the isoprene and the isoprene oxidation products factor, but we have four factors, if you look at the diurnal profiles of these individual PMF produced source factors, you see that most of the methane is being attributed to the dairy and livestock factor, with some contribution coming from the urban and oil and gas factor. This is also the season when we did have continuous nitrous oxide measurements, and most of the nitrous oxide seems to be coming from the dairy source in this seasonal period, with some contributions from the agriculture and the soil emissions source factor. Should be noted that this is the time of the year when the agriculture operations are rapidly winding down, so the use of fertilizers is not as predominant as it would be in the summer and the spring season, and we'll see that in the later PMF distributions for those particular seasons. The wind direction in this seasonal period is still fairly strongly from the west and the southwest, and that brings emissions from the upwind urban core of San Francisco Bay Area and the Rio Vista gas fields to Walnut Grove tar, and that is perhaps the reason why we see a stronger influence of the urban and oil and gas factor on the methane distribution, and this about 15 to 20% of the methane is basically coming from the urban and oil and gas source, as opposed to the dairy source, which is still the dominant source for methane in this season. If we move on to the winter season, you see that the PMF no longer produces the agriculture and the soil emission source factor, probably because there's not as much emissions from the constituents of that particular source profile. We see only three factors are reproduced. If we look at the diurnal distribution breakup, the source-wise breakup, we see that most of the methane as well as the nitrous oxide is being attributed to the dairy and livestock source factor during this time period, with a very tiny contribution, which is well within the scope of our uncertainties, coming from the urban and oil and gas factor. Should be noted that during this time of the year, the wind directions have kind of reversed. We don't see that westerly flow, daytime flow anymore. We are more looking at north-south along the valley flow, and that could perhaps be the reason why we don't see the heavy influence of the urban and oil and gas factor on the methane emissions, even though perhaps if the natural gas emissions during this period are probably higher because of increased natural gas used for space heating in the urban core. If we move on, as I just mentioned, there's a feature which you'll notice is that in any time we have the urban and oil and gas factor, you see some methane being attributed to that source factor, but we never see any nitrous oxide being attributed to that factor. Now, if the urban oil and gas factor is predominantly oil and gas, this makes sense because we would see fugitives of methane in that factor, but no nitrous oxide. It would be a little surprising if if this source factor is primarily composed of urban emissions, because you would expect some N₂O emissions from the urban core in terms of the transportation contribution or the industrial contribution to show up in this source factor, but we do not. Anyways, that's a thought I just want you to keep in mind as we look at some of the other distributions as well. Now we come down to the early part of the year, the late winter, the early spring season, late winter, and we see that now we have the agriculture and the soil emissions and delta factor being reproduced by the PMF like it was during the late fall season. And you see a contribution of methane to that particular source factor as well. We believe that this contribution of methane to this purple source factor could be due to the early planting season and the drainage that is and the heavy drainage that is applied to agricultural fields and the wetlands in preparation for the rice the rice cultivation season. And there is literature from which we have quoted in our report, which suggests a strong incidence of methane release during the early planting season from rice paddy fields. And we believe that some of this methane, which is being attributed to the agriculture source factor, is wetland related and agriculture drainage related emissions. Once again, if you look at the diurnal profile of the four sources which we four sources that we produce in this PMF analysis for this season on the bottom right, you'll see that the dairy and livestock factor is very synonymous to what primary emissions look like. The primary biogenic and secondary organics, as I mentioned before, has a diurnal profile which peaks in the middle of the day and is indicative of light and temperature dependent emissions. The black source factor, the urban and oil and gas source factor, is also primary driven, but it has a broader peak, suggesting some transport of emissions influencing the Walnut Grove tar site, as opposed to the dairy and livestock source factor, which looks more local driven. And so these are kind of the features which we use to explain what we are observing. If you look at the methane distribution, as I mentioned, we do see some methane contribution being attributed to the agriculture soil management delta factor, but the predominant emissions of methane still come from the dairy source in this season as well. We didn't have nitrous oxide measurements during this period, but it would have been interesting to see how nitrous oxide distribution would have looked like in this period. Moving on to the spring season, now we see that it's now getting warmer, and now a fair amount of isoprene is being produced by the trees and the agriculture that's coming about, and now the PMF produces a distinct isoprene related source factor. In terms of the methane and nitrous oxide distribution, we do have nitrous oxide measurements during this period, and this is when the observations get very interesting. The nitrous oxide in this season seems to be heavily influenced and attributed to the purple source factor, or what we call the agriculture and soil management source factor, while with a minor contributor coming from the manure management and the dairy and livestock factor. When we look at methane, the methane distribution looks very similar to what it looked in the period before this, which was the early spring period. So to wrap up, most of the N₂O is now being attributed to the agriculture-related factor. This is in sharp contrast with what we saw in late fall, when most of the methane was still, most of the N₂O was being attributed to the dairy and livestock factor. And if you look at the methanol distribution, diurnal distribution at the bottom right, you see that most of the methanol in this source in this period is actually being attributed to the biogenic source. Whereas in previous periods, when the biogenic source was not that strong, a lot of the methanol was being attributed to the dairy and livestock source. So moving on, summer 2013, we see now that the isoprene oxidation products are apportioned as a separate factor in the PMF analysis. In terms of the methane and nitrous oxide distributions, they look kind of very similar to the spring pattern, with most of the nitrous oxide being attributed to the agriculture and soil management factor, with a minor contribution being attributed to the dairy factor. But for methane, the story remains pretty much consistent over all the 7 PMF seasonal periods, that the methane, the primary emissions of methane, which are influencing Walnut Grove tar, seem to originate from dairy and livestock emissions, and with the minor contributions from the urban and oil and gas factor, and the agriculture and soil source as well, depending on the time of the year or the season in which we are conducting the PMF analysis. So we also see that there's some splitting of, there's a phenomenon in PMF called splitting of factors, where if you try to explain your variation by adding more and more factors, but you only have limited number of degrees of freedom to explain your factors, some of the contributions get apportioned to other factors erroneously based on the simultaneous covariances being observed in 2 factors. And we observe some of that splitting phenomena with regards to nitrous oxide in this season's PMF, which doesn't really mean that there's nitrous oxide coming from the isoprene factor or primary biogenics factor, but what it means is some of these sources are co-located, and it's not in the statistical power of PMF to explain appropriately the distribution of emissions to a single source necessarily. Summer 2012 looked very similar to summer 2013, and I just put it here for the sake of completeness. We didn't have nitrous oxide measured in summer 2012. That's why I refer to summer 2013 when we explain the summer PMF, summer distributions. What was interesting about the summer 2012 analysis was that in the urban oil and gas factor, unlike summer 2013, we had a very strong attribution to acetonitrile and acetone and propanol in that particular source factor. So we try to explain that by doing the analysis, redoing the analysis and looking at a 7-factor solution instead of a 6-factor solution. And while that was not completely successful, what we did see from the 7-factor solution was that the acetonitrile and acetone get actually attributed to a completely separate factors of their own if you look at a 7-factor solution. And when we try to explain this by looking into the vertical diurnal distribution, observation-based diurnal distribution profile of acetone and propanol, acetonitrile and for summer 2012 and summer 2013, which is basically on the plots on the left and the right, we see that in summer 2012, there was a huge influence of acetonitrile being mixed down from the top, from the upper part of the boundary layer down at our measurement level at 91 meters above ground level. And so this was primarily due to, this acetonitrile was primarily due to a biomass burn, not biomass, there was a fire, a large-scale fire called the Rush Fire, which took place in 2012 summer, and it was it's the second largest fire recorded in California's history. This is not the same as the Yosemite fire, which was the Rim Fire, which followed in 2013, but in the 2013 plot, you don't see that acetonitrile contribution here because the Rim Fire took place later in the year after this period. It was not in summer 2013, while the summer 2012 Rush Fire was in the month of July and August. Anyways, so it shows that we do detect biomass burning, or rather a biomass burning factor, and it shows strong presence of acetonitrile, the biomass burning tracer, which Alan mentioned about in the beginning, but we don't see significant contribution of methane to that source factor in light of the dominant contribution of methane, that is the domination of the dairies and livestock in the influence it has on the Walnut Grove tar. These pie charts are basically just to wrap up what we just looked at, the 7 seasons. And if you look to the plot to the left, which is for methane, you'll see that throughout the 6 seasons, 6 periods, we see that methane is primarily coming from the dairy and livestock factor. And depending on the season, the agriculture and soil emissions factor, which probably contains emissions from rice agriculture and wetland emissions, they become more prominent in the summer and spring season, and while completely disappearing in the winter and late fall periods. And we see varying contributions of methane from the oil and the urban and the oil and gas factor, which is kind of driven by the meteorology as much as it's driven by emissions. For N₂O, we see a sharp contrast in the apportionment in different seasons, with the most bulk of the nitrous oxide coming from the dairy source factor during those seasonal periods where agriculture is not a strong source, and while in those seasonal periods where agriculture is a strong source, when we're in the agriculture season or the growing season, N₂O emissions, soil-driven N₂O emissions being apportioned to the agriculture and soil management source factor becomes the predominant source of nitrous oxide as opposed to dairies. What I'll wrap up with is a quick comparison of these 6 seasonal PMF periods with the inverse analysis-based distributions which Mark conducted, broken down by seasons, and see how does for the specific region 378 surrounding the Walnut Grove Tar, and see how the PMF distributions compare with the inverse analysis top-down estimate. And what you'll see here is that what I've done for the purpose of this analysis is that I have combined the contributions in the inverse analysis from landfills, wastewater treatment plants, oil and natural gas, all into and lumped into one category, and I call that, and I basically painted in black to be consistent with what we call the urban and oil and gas emission source in the PMF distribution to the left. And what you'll see is that, in general, the seasonal trend that we observe is replicated even in the inverse analysis, although the contribution of methane to the combined source of landfills, natural gas, wastewater treatment plants, as seen in the inverse analysis, that contribution is higher for every seasonal period. This kind of indicates that the footprint which the PMF sees, or the influence which PMF analysis can detect, is kind of restricted to the region immediately surrounding the Walnut Grove Tar, and transport of emissions from the upwind urban core farther away, about 100 or 150 kilometers away from the Walnut Grove Tar, probably is not detected with the same statistical power as the influence of some of the local and closer by sources, since we don't see that much methane attributed to the oil and gas factor in the different seasonal periods on the PMF plots as we see in the inverse analysis plots, which covers a much bigger and larger footprint of 3 broad regions of California. For nitrous oxide, the same plots, and it's a much different picture here. A lot of, in the inverse analysis plots to the right, a significant amount of the nitrous oxide contribution actually comes from urban sources, transportation being a big source. There's industrial emissions, there's wastewater treatment plant emissions, and if we assume that these urban emissions or the enhancements of these urban variations are too well mixed to be detected at Walnut Grove Tar by the PMF analysis, we wouldn't see those emissions as clearly as we do in the inverse analysis, and that kind of is seen in the PMF seasonal distribution plots, where we really don't detect any contribution of N₂O in the urban and oil and gas factor in any of the seasons, and much of the N₂O is being dominated by nearby sources, agriculture and the dairy and livestock sources. So it shows some of the constraints of PMF, but this kind of VOC-based source analysis also shows the power which a greenhouse gas VOC combined source apportionment can have in terms of relative distribution of sources during different times of the year. So I think to conclude and present our recommendations for future work, I think I'll have Alan come over and wrap it up.
Thank you, Abhinav, and thank you, Mark. One more thing I just want to add about the comparison between the PMF analysis and the inverse analysis is you should recognize that the inverse analysis focuses only on the daytime data, and the PMF analysis focuses on 24-hour-a-day data. And so because of that, the footprints that are contributing to our source attribution are, again, just daytime footprints for the inverse analysis, but they're the average of all the 24-hour-a-day footprints for the PMF analysis. And I think that actually does lead to some of the differences we observe, whereby the more local sources are impacting the PMF analysis to a larger degree, and the inverse analysis is seeing the sources that are dominated by the daytime footprint and wind direction. Okay, so some conclusions. Probably the most obvious conclusion is that livestock are the largest regional source of methane, period. And that the maximum percent contribution occurs in the late fall and the winter, and that's largely because the other sources are smaller at that time. Okay. The second most important source of methane in our analysis, the PMF analysis, is the microbial-mediated temperature-dependent emissions that come from a combination of agriculture, soil management, and the delta wetland ecosystems. And those are, again, highly seasonal and low in the fall, winter. The third urban and oil gas source contain methane, but I think it's important to state that we didn't see the N₂O in that source in our PMF analysis. And that's from an aggregation of the upwind sources in the San Francisco Bay Area and the Rio Vista natural gas fields. So with respect to N₂O, our PMF analysis really only discerned two significant source categories. And one of those was the ag and soil management and delta source, that microbe-driven source that correlated with the seasonality of agriculture and fertilizer use. So it was dominant in spring and summer. And then the dairy and livestock manure management source, which was occurring all year, but it provided the dominant contribution in late fall and winter, basically because the other source was so much lower in that season. So we conclude that seasonality has a very strong influence on the methane and N₂O biological emissions, and this phenomenon is clearly observed using this top-down approach and inversion modeling. So some recommendations for future research. There are two conclusions from this work that we think need further investigation. One is the seasonality of the major sources of methane and N₂O from the agricultural sector. And that seasonal variability has real implications for how past studies and current studies should be used or not used for defining the emission inventory. We have to recognize that a short-term study in a period where those emission sources are high doesn't represent the whole year. And so we really have to start thinking about studies that are going to integrate over the full year. So we were recommending a network of long-term ground-based measurements of concentrations and fluxes, so we actually get the emissions combined with modeling to define the seasonality from specific source categories. And there are projects now funded by CARB and internal work at CARB that share these goals and are underway, but we think this needs to be a long-term sustained effort because we need to know whether these sources are changing through the year, but then how they also change from year to year. You know, if we're going to be able to confirm that by 2020 we're down to 1990 emission levels, we need to know how these things are changing with time, not just a snapshot of what they look like now. What we've done thus far is 1 year trying to provide a snapshot of what that year looks like. It's actually much harder to define the absolute emissions in a given year than it is to look at a continuous data set and define changes in emissions from one year to the next. So I'd strongly encourage the Air Resources Board to try to develop and maintain those longer term records that are going to be able to show us whether things are changing from one year to the next. That's the only way we're really going to know if the reductions in the emission inventory can be confirmed by field observations. Second point, the magnitude of the N₂O emissions from the transportation sector. In the inventory for California, it's that 12% of the N₂O is from the transportation sector. I can only say that we don't see evidence for that in our PMF analysis of the observations at the Walnut Grove Tower. We also did not see evidence of that in the PMF analysis that Abhinav referred to that was done previously in the Bakersfield area during the CalNext 2010 study. So what we've done with the PMF analysis of VOCs and greenhouse gases is inconsistent with the current inventory. And we think this deserves some further study. And again, this is a topic that CARB is already working on. And I think we should try to get to the bottom of that, because the issue of whether vehicles are contributing is important. And also, since the emission control strategies on those vehicles are changing with time for the fleet, that contribution is also likely changing with time. So it deserves some attention. Finally, I would say that our work has developed a baseline understanding of how to use the VOCs as greenhouse gas source tracers, doing this kind of top-down PMF analysis. And we recommend that ARB continue work in this area and invest in further applying and developing this approach. And a critical next step would be to add more tracers. So we limited our analysis to those things that we could measure with the PTR MS. It was about 20 different tracers that we kept track of for the year. We recognize that it would have been extremely helpful in particular to have measurements of alkanes from ethane on out to pentane or hexane that would have been more useful indicators of the types of fossil fuel related sources that were contributing methane. Also, the technology for making these kind of measurements is advancing rapidly, and I think there are really good opportunities for doing studies with more tracers. The more tracers that we have, I'd say the more representative the PMF analysis can be of specific sources. So with that, I would like to thank our whole team and invite questions. Thank you for your attention. And I'll also say that questions should be directed to any of the three of us. So maybe the other two can also come up for this as well.
Marc Vessier, ARB. I am curious about the apportionment with the PMF, about those sources that are coming from wastewater treatments and the landfills. Obviously, there are also some anaerobic fermentations. Wastewater management may not be very distinguishable from manure management. So are they being lumped more with the dairies, or are they lumped more with the ag management and Delta?
It's a really good question. So first, I don't think there's a lot of wastewater treatment emissions in that particular area, and I'm looking at Mark to see if I've got that right. But we wouldn't expect, based on the, let's say, the a priori maps of emission sources, that those would contribute significantly in the region. So it's not surprising to me that we couldn't pull it out. But again, with the PMF analysis, we're only going to be able to pull it out if there's a specific chemical tracer or two that we can measure that are coming from that source and not others. On the other hand, I was a little bit surprised that we maybe don't see more from landfills, but the landfill emissions, they're very clearly point sources. They're large point sources, And there aren't that many really close to the tower, but there are some in the region. So in the inverse analysis, you do see a contribution from those, but we didn't see any particular indicators that would tell us about the specific landfill emissions. However, I think that's something we could work on in the future. And one example of something I expect to come from landfills and wastewater treatment facilities both, it turns out to be chemicals called methylsiloxanes, cyclic volatile methylsiloxanes, which are dominant compounds in antiperspirants and other personal care products. They get washed off people and go into the wastewater treatment plants, and it turns out there are major emissions of those from wastewater treatment plants. Also major emissions from landfills as those consumer products go into the landfill. And with the PTRMS that we had out at the site, we couldn't have seen those, but we did do a little bit of pilot measurements with a newer version of that instrument during this study called a PTR TOF-MS. It's a time of flight mass spec instead of a quadrupole. We can see a lot more tracers, and we were actually able to see those cyclic methyl siloxanes with the PTR TOF-MS, and we think they're probably indicators of those two types of sources specifically. But that's just one example. And so in order to differentiate more and more sources, we would need more specific tracers. And I think it's a good goal, but that we couldn't do it, we couldn't see it based on the data that we had at this point. Okay, more questions? Oh, okay, so the question is more specific differentiation possible within livestock emissions? That's a really interesting question. I think from the PMF analysis at the larger scale, unlikely, because livestock emissions are generally co-located. However, if we go into a dairy or livestock area, we can look more specifically at processes that are happening there. And if we found that there were differences in the tracers that, say, come from the waste lagoons versus directly from the cows versus other ways of managing their waste, for example, perhaps we could start to differentiate. But based on the tracers that we had with the PTRMS measurements, I don't think so, because all of those sources within the dairy produce a lot of the tracers that we were looking at. So methane, methanol, some N₂O, acetaldehyde, et cetera. So I don't think we had tracers that were specific to different processes within dairy or manure management. However, that doesn't mean it couldn't be done, but we'd have to think hard about what those emission profiles look like and then make sure we had those tracers measured continuously at the tower at the same time. Chris?
Thanks, all three of you, for a great talk. You mentioned that a lot of the methanol has a biogenic source. You also mentioned toluene also having a biogenic source. I was just wondering if you've looked into what percent of the toluene might actually, or the seasonal pattern in biogenic toluene.
So there is some evidence for biogenic toluene. And on a global scale, the biogenic toluene may rival the toluene from anthropogenic sources, but anywhere near activities that are urban or with oil and gas, et cetera, the toluene is going to be completely dominated by anthropogenic sources. And so the patterns that we observe in the data at Walnut Grove, I believe, suggest that the toluene is anthropogenic, that we can tie it to anthropogenic sources. We do have some measurements up in the Sierra Nevada mountains suggesting that there's a little bit of toluene actually coming from the forests up there, for example, but it's a relatively minor source compared to toluene from anthropogenic sources, particularly if you get anywhere near oil and gas fields, vehicles, urban sources. Yeah.
Can I quickly add to that?
Yeah, I mean,
I can quickly add to that. So the tidal, Hello? So so this diurnal profile of benzene, toluene, and carbon monoxide during most of the seasons looked very similar, except for summer, when the toluene diurnal profile was distinctly different from benzene and CO, which were, by the way, similar to each other. And if you look at the PMF analysis, there is some of the toluene does get apportioned in the summer to not just to the urban and oil and gas factor, but to the agriculture and soil emissions factor as well. And that kind of led us to introduce that idea, even in the text of the report, that there is some element of some contribution from biogenic toluene in the summer season. But that goes away during different other times of the year. Yeah.
Thanks, Abhinav. Yeah, Klaus.
Quick question about acetonitrile. Is that associated more with flaming phase of biomass burning or smoldering?
That's a really good question. I don't know the specific answer except to say that acetonitrile is generally used as a tracer of burning biomass, and I don't know how the emission ratio of, say, acetonitrile to CO, CO2 changes as a function of the stage of the fire, or flaming versus smoldering. I think that information is known. I just don't have it at the tip of my mind.
Okay, if that takes care of the questions for the moment, I'd like to thank all of the audience, both here and on the web, and I'd like us to thank our speakers again.
Thank all of you for your attention, and sorry we went so far over.
Okay, Marco.